So, good day everyone. I'm your reporter for today, Abigail Limliman. So, our topic is all about genetic cloning. So, before that, alamin muna natin kung ano yung genetic cloning or tinatawag nating cloning. So, yung cloning refers to the process of creating an exact copy of a biological unit. So, yung cloning is um, nagke-create siya ng exact copy ng isang biological na katangian or isang tao. So, the birth of the cloned sheep Dolly in the United Kingdom in July 1996 attracted the attention of the world with the successful application of a cloning technology using the nuclei of mammalian cells, which had previously been considered to be impossible. So, yung The Birth of the Cloned Sheep Dolly sa United Kingdom noong July 1996 is naka-attract siya ng attention ng world para maging successful yung kanilang pagko-cloning sa sheep Dolly. At ang ginamit nilang technology is yung nuclei of mammalian cells, which had previously been considered to be impossible. So, cloning technology is widely expected to contribute to the stable supply of livestock food sources, to the manufacture of pharmaceutical products, to the protection and reproduction of rare animals, and to mass production of test animals. So, yung mga cloning technology, ine-expect na mako-contribute siya sa iba't ibang supply ng um, iba't ibang supply, and then ng iba't ibang livestock food sources na iba't ibang technology ng mga products ng mga um, tinetest nilang animals. So next, in Japan as well, in September 1997, the Council for Science and Technology established the Bioethics Committee to engage in studies into the issue, including hearings for gathering public comment from the people. So, sa Japan, noong September 1997, yung Council for Science and Technology is nag-establish sila ng bioethics para ma-engage yung studies na sa mga issues sa including doon yung hearings for the gathering public comment from the other people. So the result was the enactment and promulgation of the law concerning regulation relating to human cloning techniques and other similar techniques. So yung result ng enactment na yon sa law concerning regulation relating to human cloning techniques and other similar techniques in December 2000. So, this law bans the transfer of human cloned embryos, etc. into human or animal wombs for the purpose of producing human cloned individuals and stipulates severe penalties for violators. So, sa law na concerning regulation relating to human cloning techniques and other similar techniques, nagban sila na kapag nag-transfer ka ng mga human clone embryos sa mga tao or sa animal wombs, magkakaroon ka ng penalties kasi na-violate mo yung law na yun. So next is, in addition, with regards to the creation of human cloned embryos, the law calls for the Council for Science and Technology policy to engage in discussions about the future handling or fertilized human embryos while banning the practice for the time being in accordance with guidelines established under the law. So in addition, yung law na sinabi kanina is nag-regard sila ng kapag nag-create ka ng human cloned embryos, yung matatawag na locals is yung Council for Science and Technology policy para ma-engage na ma-discuss about yung future handling of the fertilized human embryos para ma-discuss or alam pa natin yung kung ano yung mga gagawin between fertilized human the embryos. So, sabi naman ni Binoaji 2000 defined cloning as the isolation and proliferation of individual genetically unique cells. It is a kind of high-resolution separation method for DNA molecules which could be difficult to resolve by any other means. So, ayon naman kay Binoaji nung 2001, dinedefine niya yung cloning as the isolation and proliferation of the individual. So, itong kind na high resolution na to is yung pagkuklone ng ng mga embryos or mga human is sineseparate siya from the DNA molecules which could be difficult to ma-resolve ng other physicians. So next, yung the word clone daw was first used to describe a population of cells or organisms all derived from a single cell or organism by asexual multiplication. So yung clone daw was first used para ma-describe yung population of the cells and yung mga organisms, and it is all derived from a single cell or the organism by the asexual multiplication. So all members of the clone look exactly alike since they possess the same genetic constitution. So lahat ng members ng pagko-clone is look exactly alike daw kasi na-process nila ng mabuti yung genetic constitution. So the birth of recombinant DNA technology made it possible to clone a specific gene or a segment of it in bacteria. So nung the birth of recombinant DNA technology nagawa nilang posible yung pagkuklone ng iba't ibang other specific na bacteria or kung ano man para makagawa ng isang ka-lookalike niya. So this procedure was appropriately termed gene cloning. Later, it also became possible for new combinations of unrelated genes to be constructed in the laboratory, so the genes from the different organisms or species could be spliced together in vitro to create new recombinant products. So sa paggagawa ng sa pagkukuha ng mga sa paggagawa ng cloning is kailangan nila ng mga laboratory para sa mga nakuha nilang genes and sa different organisms or species para makapag-create or makagawa ng bago and magandang klase ng lookalike ng isang tao or yung animals. So next is the cells which contain the recombinant DNA molecules with the gene of interest is then selected and separated from the remaining cells that were not selected. The selected cells can then be amplified many times. So yung mga cells na nakukuha nila sa mga ginagawa nilang magka-lookalike is nagiging amplified kasi ginagamit siya many times. So during this process, the recombinant DNA molecules will be replicated and expressed in the whole cells. So this technique is useful for the synthesis of large quantities or scarce proteins such as human insulin or growth hormone. So during sa process ng mga paggagawa ng recombinant DNA molecules, nagiging replicated and expresses siya sa mga host cells. So, ang technique doon is para maging useful sa mga gagawing ka-lookalike nung isang gagawin nilang cloning. So, moreover, gene recombinant technology, which is already in technical use in society, is well suited to elucidation of the framework of living things, to the efficient manufacture of medical and pharmaceutical products, and to the improvement of agricultural crops, and etc. And expectation are high for its technological application. So, sa gene recombinant technology, which is already technical use technical use kasi nagagawa ng maayos and maganda yung mga products ng isang pharmaceutical products, and nag-i-improve siya ng mga agricultural crops at mga iba pa. So, nung 2000, the Cartagena Protocol on Biosafety to the Convention on Biological Diversity was adopted under the auspices of the Convention on Biological Diversity. So, sa purpose nun of pag-a-adapt nila is to measure in line with the safe transport, handling, and utilization with particular focus on the appropriate control of movement that crosses international borders. So, noong 2000, yung Cartagena Protocol on Biosafety to the Convention on Biological Diversity is na-adapt sila ng auspices of yung Convention on the Biological Diversity para sa purpose nilang pag-adapt nun is to measure the line with the safety transport, handling, and utilization with the particular focus para ma-control yung movement na pinaggagawa ng mga international borders. So last is in Japan, the law for preservation of biological diversity through regulation of the use, etc., of gene recombinant life form, etc., stipulating the domestic measures required to conform to the protocol was passed in June 2003. So, yung sa Japan, yung law na preservations of biological diversity, tapos yung gene recombinant life forms is nakapasa noong June 2003. So, that's all my report. Thank you for listening.